Okay, chapter 8.2 is all about cell respiration. So here's some big mark questions that we want to make sure we're able to answer by the end. So outlining the process of glycolysis, you're going to see this command term a lot from this chapter just because there are so many processes, and outline just kind of lends itself well to knowing what the steps are. Describing the processes that occur in the mitochondria when oxygen is present. That sounds a little tricky. And then explaining how chemical energy is generated by the electron transport chain and chemiosmosis. Before we can start talking about anything having to do with metabolism, particularly in the mitochondria, we need to talk about something called a redox reaction. So redox comes from reduction and oxidation reactions, and they're always happening together, so they happen simultaneously. Oxidation means the loss of an electron. So if something loses an electron, it is said to be oxidized. If something gains an electron, it is said to have been reduced. So this purple guy over here is stealing an electron from the yellow guy. So the purple guy gains it, he gets reduced, and the yellow guy loses it, he's going to get oxidized. Remember that going along with electrons is energy. So sometimes it's hard for me to imagine what is gaining or losing electrons, but I can think of what is gaining and losing energy pretty easily. So in cell respiration, we're breaking down glucose and we're using the energy in the bonds of glucose to make ATP. So we're stealing the energy from glucose. That means glucose is what's going to get oxidized. Okay, so just kind of a general reminder there. You're going to hear me say oxidation and reduction a lot. There are a lot of questions revolving around this concept, so make sure that you understand that. Another thing that we want to be mindful of is stuff from standard level. So we often see these concepts paired together in section B on paper 2, where you have those like really fun 3-part questions like A, B, and C, and you think it'll be all higher level stuff, and then they throw some standard level concepts in there just for fun. So to make sure we all know what we're talking about here, glucose, which is a 6-carbon sugar, gets broken down into 2 molecules of pyruvate. Each of them have 3 carbons, and that process yields to ATP. This is going to happen anaerobically because it's in the cytoplasm, so we don't need any oxygen yet. From there, it's kind of like a choose your own adventure kind of situation. If oxygen is present, we undergo what's called aerobic respiration. All organisms produce the same products after aerobic respiration: water, carbon dioxide, and then a whole bunch of ATP. So that's kind of the ideal scenario, is aerobic respiration. It produces a lot of ATP. If there is no oxygen present, then this process looks different depending on what kind of organism you are. So I should go back and tell you, aerobic respiration happens in the mitochondria if you're a eukaryote. And then if you don't have oxygen present and you have to do fermentation, you're going to stay in the cytoplasm. So no mitochondria for you. Now, fermentation is going to take pyruvate and it's going to break it down into different products depending, again, on what kind of organism you are. Animals like you and me and most bacteria are going to produce lactate, so they're going to turn that pyruvate into lactate. Yeast, on the other hand, are going to turn that pyruvate into 2 products, alcohol and carbon dioxide. So, tricky question that comes up a lot is about yeast, because yeast will produce carbon dioxide whether they're fermenting things anaerobically or aerobically. So you have to kind of be careful. You got to look at what else they're producing, whether it's water or whether it's alcohol, or you can look at the ATP yield. So aerobic always produces more ATP. Fermentation doesn't get any additional ATP. All you get is this kind of like lame 2 ATP that you made up here with glycolysis. So just watch yourself there. All right, so now for the fun stuff. Before we get real detailed, I just want to give you an overview of where we're at, okay? So we're going to talk about several different steps in cell respiration, and we're going to talk about them in depth and separately, but they are all part of the same big picture. So glycolysis out here in the cytoplasm, okay? Then something called the link reaction, the Krebs cycle, aka citric acid cycle, and the electron transport chain and chemiosmosis also happening in here in the mitochondria. So just make sure you understand that these processes are all related, even though we're talking about them separately. For glycolysis, again, we are in the cytoplasm, and we are going to start out with a 6-carbon glucose molecule. So we're going to kind of abbreviate that as 6 circles drawn together, and we're not going to bother with the oxygens and the hydrogens because it's all about the carbons. All right, well, to get glycolysis started, we're going to have to invest some energy, and that energy is going to come from ATP. So we are going to steal 2 phosphates, one from each ATP, all right, to do what we call phosphorylation. So we are sticking some phosphate groups on the end of this glucose molecule. You don't have to know the name for this intermediate molecule, but what this does is it makes this unstable. So phosphorylation requires some energy. Again, these phosphate groups are coming from the ATP, and the point of that is to make it unstable. The next phase is called lysis. Lysis literally translates into to break. So we're going to take that glucose molecule that has now been phosphorylated, and we're going to break it in half. An inorganic phosphate from the surrounding cytosol is going to be able to join on the other side here. We don't need ATP for this. We have a missing bond here, so no more ATP, just an inorganic phosphate group. So there's little antler-looking things on the side of my 2 molecules. Then we're going to go through a phase called oxidation. So whenever we have oxidation happening, reduction is happening at the same time. If something is losing electrons as an oxidation, then something else has to be gaining those electrons. So this is the molecule that is getting oxidized. We are going to steal electrons and steal energy from this molecule. And we're going to use those electrons and energy to reduce NAD into NADH. Okay, and I'm going to get 1 of those per molecule. So 2 NADs get reduced into 2 NADHs. And then we're also going to be able to use each of these phosphate groups to rephosphorylate ADP into ATP. So I'm going to do that 4 times total. 2 for this molecule, so 1 ADP to ATP here, another ATP here, another ATP here, and another ATP here. So I'm going to have, at the end of this, 2 molecules of pyruvate. They have 3 carbons each. I started with a 6-carbon glucose. That has now been cut in half, so 3 and 3. I've also stolen some energy from that glucose. Glucose has been oxidized, and I've used that energy to make these high energy molecules called NADH and ATP. And actually, I'm looking at this here and I see a problem in my own answer key down here. So I'm not making 2 ATP. I need to say that I have a net gain of 2 ATP. And that is because we had both an energy investment phase and an energy harvest phase. So in the beginning here, I spent 2 ATP. All right, I made 1, 2, 3, 4. So I made 4 ATP. That gives me a net gain of 2. So I would say in general, when you're talking about glycolysis, try to be consistent with your wording in saying a net gain of 2 ATP, just so that the reader can be sure that you understand that you had to spend some and then you made some also. All right, so here we are again in our overview. We just talked about glycolysis. We were taking that 6-carbon glucose and turning it into 2 of these 3-carbon pyruvates. I was able to make a net gain of 2 ATP, and that's something that we call substrate level phosphorylation. It means I'm just adding it on there. We'll talk about this other mess later, okay? And then I made a few high energy electron carriers called NADH. NADH is the reduced form, okay? And I was able to reduce that because I oxidized glucose. Now, let's take that pyruvate that we just made, and my eventual goal is to take that pyruvate and put it into the Krebs cycle over here. But I can't just go directly there. It doesn't work that way. I have to first go into what we call the link reaction. And the link reaction is what we are going to study next. So by the end of the link reaction, we are again going to oxidize this pyruvate. We're going to steal some energy, and we want to turn it into a compound called acetyl-CoA. I have the link reaction here twice. I personally don't care if you draw it twice or not, but the reason why I drew it twice is because at the end of glycolysis, we had not 1, but 2 pyruvate molecules. So here's what's happening in the link reaction. That pyruvate is going to get decarboxylated. So carbo kind of reminds me of carbon. D means to take something off, like decapitate would be to take off your head. That's bad. Okay, so here I'm not going to take off your head. We're going to take off a carbon. So when we take off a carbon, that carbon gets released as carbon dioxide. So watch how that happens here. Here's my 3-carbon pyruvate. If I pluck one of these carbons off, it gets released as carbon dioxide. And then I no longer have 3 carbons, I have 2. Now, whenever you break the bond, okay, between 2 carbons, that's a high energy bond. Breaking that bond is going to release some energy. And I'm going to use that energy, that electron that they're sharing there, to reduce NAD to NADH. Okay, so pyruvate is going to get oxidized. We're going to steal some of its energy, and NAD is going to get reduced into its reduced form called NADH. Okay, so at the end of this, okay, I should only have these 2 carbons left. Yep, here they are. But to enter the Krebs cycle, they need a little chaperone, and that chaperone is called coenzyme A. So in addition to this 2-carbon compound, we are going to attach coenzyme A, and that is how we're getting this compound called acetyl-CoA. So acetyl-CoA is these 2 carbons left over from pyruvate plus the coenzyme A. So if we ignore my written over copy, here's what we're kind of starting with. We're starting with a pyruvate molecule, and I'll show you where that is. That is right here. And then we're going to end up with 2 acetyl-CoA molecules, 1 for each of those pyruvates that I started with, 2 carbon dioxide molecules. So for each carbon that we plucked off, we're going to be breathing out a carbon dioxide molecule, and 2 NADHs. So 1 for each of those pyruvates. So again, pyruvate is going to get oxidized and NAD gets reduced into NADH. And that is the link cycle, or the link reaction. These NADH guys, we're not going to use those until later. They'll come back into the story in a little bit. For now, we're going to focus on what happens to these acetyl-CoAs. So again, here's our overall picture. Okay, we just took pyruvate and we oxidized it. Okay, and we are now ending up the link reaction with acetyl-CoA. Acetyl-CoA is going to enter what we call the Krebs cycle. Some old school people call it the citric acid cycle. You can call it either. That's totally fine. All right, and we're going to see how acetyl-CoA changes in that process. So we are now fully into the matrix of the mitochondria. The Krebs cycle, again, we're in the matrix of the mitochondria, is going to start with that 2-carbon acetyl-CoA. I'm only going to show one of them, okay? So we'll have to remember that for every molecule of glucose, the Krebs cycle happens twice, but I'm only going to show one. That acetyl-CoA compound is going to get picked up by a 4-carbon compound called oxaloacetate. You don't have to know the name of that. That's fine. I'll tell you what you have to know about that compound in a little bit. When oxaloacetate, this 4-carbon compound, picks up the 2 carbons from acetyl-CoA, it becomes a 6 carbon compound. 2 carbons from my glucose molecule, okay, so from my original glucose, and then the 4 carbons from the oxaloacetate. Now that we're fully in the cycle, we don't need this chaperone. We don't need this coenzyme A anymore. So as this oxaloacetate picks up these 2 carbons and becomes one giant molecule, that acetyl-CoA is no longer acetyl-CoA. That coenzyme A comes off. The next step is to decarboxylate that molecule. Okay, so again, we had a 4-carbon molecule plus a 2-carbon molecule gave us a 6-carbon molecule. Sometimes in diagrams you won't see these circles. It'll just say 6C, and you have to know what you're looking at there. So when I pluck one of those carbons off, it is now a 5-carbon molecule. And again, when we decarboxylate something, it comes off as a carbon dioxide molecule. When you decarboxylate something, that is going to, again, break a bond. So when we break a bond here, that compound is going to get oxidized. It's going to lose the electrons that it's sharing here. So if this compound is getting oxidized, that now means that I have the energy and the electrons to reduce something else. Okay, and so I'm going to take NAD and I'm going to reduce it into NADH. So this compound is going to get oxidized and this compound gets reduced. So the carbon compound is losing electrons, losing energy, and NAD is gaining electrons, gaining energy. And I'm going from a 6 to a 5. Okay. We're gonna do that all over again. So we're gonna decarboxylate another end of our molecule here. So this carbon is gonna come off to form carbon dioxide. And that also, again, releases some energy. So this compound is going to get oxidized. And I'm going to be able to reduce NAD to another NADH. So are you seeing a pattern here? Whenever a carbon comes off, I produce carbon dioxide and I can reduce the compound here. Okay, so I can make reduced NADH. You've probably noticed that at this point, I'm already rid of the 2 carbons that were part of my original glucose molecule. So these are from the original glucose. They're already gone. They're here and here. I'm left with the 4-carbon compound that I started with. So I've plucked all of those carbons off of that glucose. Well, what we have to remember is that this is a cycle. And so that 4-carbon compound is going to remain in the cycle. They don't come apart. Now, it doesn't just walk itself on over there. This carbon compound still has some residual electrons from when it was bound to these acetyl-CoA carbons. So we can further oxidize this compound and reduce a few other compounds. So this carbon compound is going to get oxidized. Notice it's not losing another carbon. It's going to lose electrons, which we can't see. Okay, so this is going to get oxidized, and I'm going to reduce NAD to NADH. I'm also going to be able to reduce another electron carrier called FAD to FADH2. So again, oxidizing this one, reducing this one. And the last thing I'm going to be able to make here is some ATP, okay? So again, this must have held quite a bit of energy in it, because even without pulling off another carbon, I was able to make all of these compounds. What we need to remember here is that all of this in the Krebs cycle is going to happen 2 times. That's because from my original glucose molecule, I was able to make 2 acetyl-CoAs. So one 6-carbon glucose molecule was able to make 2 3-carbon pyruvates, which was able to make 2 2-carbon acetyl-CoAs, which is right here. So this whole process is able to go around twice. And that's going to affect the final outcome of this, okay? So again, Krebs cycle, we're happening in the mitochondrial matrix. We're starting with 2 acetyl-CoAs. I mean, really 1 acetyl-CoA, but the Krebs cycle is turning twice, okay? We're gonna end up with 4 carbon dioxides. 1, 2, and then when it goes around, 3, 4. We're gonna end up with 1, 2, 3 NADHs, again, times 2, okay? An FADH2 times 2, and an ATP times 2. So if we think about our original glucose molecule again, okay, I had 6 carbons. 2 of those carbons came off when we did the link reaction, okay? So the link reaction released a carbon dioxide molecule for each molecule of pyruvate. And now the Krebs cycle is producing another 4 carbon dioxides. So I no longer have any carbons left from my original glucose. All of them have been plucked off and decarboxylated. So how come we're not done yet? How come we still have another step to do? Well, that's because all of these high energy electron carriers are going to be carrying the energy in that original glucose compound to the next step. So even though I don't have any more of these compounds yet, or again, the energy that was in their bonds has now been transferred to the high energy electron carriers, and they're carrying that energy to the final step. It's worth noting that you will never ever have to draw the Krebs cycle. I obviously didn't tell you that before I made you do it on your notes, but drawing the Krebs cycle is not something that you have to do. However, I recommend knowing how to draw the Krebs cycle, because you're going to get questions like this that are asking you what in the heck is going on here. So again, not showing the carbons, but saying I'm going from a 5-carbon to a 4-carbon molecule. And that's, again, process 2. So I want to be looking in this column over here. If I'm going from 5 to 4, that must mean it is being decarboxylated. Now, it is also being oxidized. It is losing energy. So I have to consider that as well. Now, because the 5-carbon compound is being oxidized, that means NADH is being reduced. So oxidized, reduced. So this one doesn't make sense. So I'm going to eliminate that for now. Over here, later on in my cycle, the 4-carbon compound is turning back into that oxaloacetate that we talked about that begins the cycle. And so this is getting oxidized so that NAD can get reduced into NADH. So let's figure out what's going on here. We already know this isn't an option. All right, NAD is being decarboxylated. Well, NAD doesn't even have a carbon, so that doesn't make sense. So then we need to think about what's happening here. NADH is the product. It itself isn't being reduced. NAD is being reduced into its reduced form, which is NADH. So the answer here should be D. Holy smokes. So again, it's helpful to know what the Krebs cycle looks like. Another possible question is you might get a picture of the Krebs cycle, of course, without any of my crap on here like that. And you may be asked to label where decarboxylation is taking place. So you would want to be able to label any spot where you're losing a carbon. It's also helpful to know how to draw it if you're answering an open-ended question about the process itself, because it reminds you of everything that you have to write about. All right, so here again, we just finished this Krebs cycle, the citric acid cycle. So we actually don't have any more carbons left. This last and final step is going to be all about those electrons that we've been stealing. So part of the NADH that we've made in the various steps, all of those are going to come over here to the electron transport chain and chemiosmosis. And actually, I'm not a fan of this diagram because we made an NADH, a couple of them actually, in the link reaction, and they forgot them. Boo. Okay, so that's what we're ending up with. All of the energy from our glucose molecule is now in these couple of lame ATPs, but mostly carried in these electron carriers that are going to help us out with the final step. All right, so far, the reactions in the mitochondria that we've talked about, like the link and the Krebs cycle, have been taking place in the matrix of the mitochondria in here. The electron transport chain and chemiosmosis, they're not really separate processes, they happen together, and they're going to happen on these folds of the inner membrane called the cristae. So in this diagram, it's kind of important to understand what in the heck we're looking at. Now, IB will sometimes give you a diagram that looks like this. Sometimes they'll flip it upside down. So here's how you can orient yourself in terms of a diagram. I want you to really watch where hydrogen ions are flowing back through this big hunker called ATP synthase. So ATP synthase is the name of the enzyme that is going to facilitate the conversion of ADP into ATP. So find ATP synthase, wherever that may be in your diagram. Look at the direction through which hydrogen ions are flowing through it. Wherever they are going into is the matrix, and wherever they're coming from is the inner membrane space. So in your diagram, if you have one, make sure that you have kind of labeled that. So again, hydrogen ions are going to flow through ATP synthase from the intermembrane space into the matrix. So as you're kind of annotating what's happening in this diagram, you can just kind of listen to me and write down whatever you need to. So these high energy electron carriers, NADH and FADH2, are going to come to the electron transport chain. And again, this is embedded here in the membranes of the cristae. They are going to donate their electrons. So what does that mean? Well, that means NADH is going to get oxidized, and the protein that receives its electron is going to get reduced. So it donates its electrons to one of the carriers in this little complex of proteins here called the electron transport chain. So this is the electron transport chain. So again, NADH gets oxidized, this protein gets reduced. A series of redox reactions is going to occur because this protein is going to donate the electron to the next one in the chain, who's going to donate it to the next one in the chain, so on and so forth. Every time something receives an electron, that is getting reduced, and whoever is giving up or donating the electron is getting oxidized. I hope you understand that, because you can't ask me questions because I'm in a video. Now, when that electron gets passed from carrier to carrier, not all of the energy that's associated with that electron is going to be harnessed either. So some of that energy is going to be lost to the electron transport chain, but not lost in the fact that we can't do anything useful with it. In fact, something really useful happens. Some of the energy from that electron is going to be used to pump actively hydrogen ions from the matrix into the intermembrane space. So here's what's happening. I'll show you over here on this diagram. We are going to be actively pumping those hydrogen ions from the matrix into the intermembrane space. Okay, now the intermembrane space is really skinny, so we're able to develop a high concentration of hydrogen ions really quickly in here. We then allow those hydrogen ions, aka protons, to diffuse passively through ATP synthase. So ATP synthase is kind of cool. It's my favorite enzyme because it does two things. It acts as a channel for facilitated diffusion, but it is also an enzyme. So as these hydrogen ions are flowing, again, passively through here from high concentration up here to low concentration back in the matrix, as that's happening, okay, this little part of the enzyme is acting like a turbine. It's actually spinning around. And that kinetic energy is enough to catalyze the reaction from ATP or ADP into ATP. Okay, so that's what those hydrogen ions are doing. Now, again, that pumping all comes from the passing of this electron, but each time we pass that electron, it loses energy. And by the time it's at the end of the electron transport chain, that electron has very little energy left. But we need to get rid of it. So who can you find that loves electrons so much they don't care whether they have energy or not? Well, that is the job of oxygen. So we haven't talked much about oxygen yet. Oxygen is needed for aerobic cell respiration for this step and this step only. It is the final electron acceptor of the electron transport chain. So NAD and FADH2 donate their electrons to the electron transport chain. It gets passed from carrier to carrier, releasing enough energy to pump those hydrogen ions. And then at the end of the chain, oxygen is that final electron acceptor. It is very electronegative. It'll take an electron no matter what. Now that it has this extra electron, it's enough to form some covalent bonds. So in here in the matrix, again, we've got all these hydrogen ions. We've been using them for pumping. We're also going to combine them with this oxygen that now has some electrons for bonding. And what do you get when you combine an oxygen and two hydrogens? Why, that would be water. So when we say that cell respiration takes glucose and produces carbon dioxide and water, I hope you're now understanding where that's coming from. The water comes from this step. It's a product of that oxygen accepting that final electron. And the carbon dioxide comes from all of those decarboxylation steps that we talked about in the link and the Krebs cycle. Okay, so at the end of cell respiration, oxygen is the reduced molecule. It gains that electron, and glucose got oxidized. We stole all of its electrons, and they end up here. All right, so just to kind of summarize here, the electron transport chain in chemiosmosis is all happening on the cristae, and the reason why these cristae are kind of foldy like this is to increase the surface area so that we can have more electron transport chains and generate more ATP. So the energy for this step comes from those high energy electron carriers, NADH and FADH2. You don't need to know the numbers, that's okay. You can backtrack and find your numbers from the link and the Krebs cycle. And of course we need oxygen to be that final electron acceptor. We're gonna end up with water, okay? And that comes again from this oxygen step, and then a whole bunch of ATP, okay? So that is what happens in the electron transport chain in chemiosmosis. You will almost certainly see a question about the direction of movements of these hydrogen ions. And again, sometimes IB will call them hydrogen ions, sometimes they will call them protons. It is the same thing. Anywho, so they are actively pumped out of the matrix into the intermembrane space, and that is because, again, that intermembrane space is super small, so I can very easily establish a high concentration gradient. That is the active transport part, okay? They diffuse passively back through ATP synthase, okay, back into the matrix. So again, pumped out into the intermembrane space, that is active transport. They flow passively back into the membrane or back into the matrix through facilitated diffusion. So I would definitely make sure you know your transport mechanisms there. Right, so speaking of the mitochondria, it's always a good idea in biology to kind of keep an eye on this theme of structure and function. So the mitochondria looks like this because each of these structures has a particular function. So let's start with this outer membrane. So the outer membrane contains the transport proteins for being able to get that pyruvate into the mitochondria for the link reaction. It also separates it from the rest of the cell. The inner membrane, which again, I'm going to trace here in red. Here's the inner membrane. Woo! Contains the electron transport chain and ATP synthase for that step that we call oxidative phosphorylation. That's just a fancy word for chemiosmosis and how that ATP is generated. The cristae are these folds in the inner membrane. So here's a cristae, etc. It increases the surface area so that you can have more electron transport chains. Oh, that's really messy. All right, the intermembrane space, and I know you've heard me mess this up because it's really easy to do. Sometimes it's really easy to call it the inner membrane space. Bad, don't do that. Inter means between, as in the space between the inner and outer membranes. So that intermembrane space is really small and skinny so that, again, we can accumulate a high concentration of those hydrogen ions, building up that concentration that we need to get them to passively diffuse back in through ATP synthase. And then the matrix, this big ol' area in the middle here, has all the enzymes that we need for things like the Krebs cycle. Let's go back to those questions that we've been kind of mentioning here. So outlining the process of glycolysis, we want to talk about the steps. So it occurs in the cytoplasm. Again, when you're writing these open-ended questions, if you're tempted to just start with the steps like I am, don't forget, fun facts count too. All right, so a hexose, that is fancy word for glucose. You're going to get marks for saying glucose. Just say glucose, all right? Is phosphorylated using ATP. So that's that first step where we add the little horns, those phosphate horns on there. That is split into 2 triose phosphates. Okay, this is looking kind of wordy. This means 6 carbon sugar is split into 2 3 carbons with a phosphate on it. It is okay if you say 3 carbons, for goodness sake. Oxidation by removal of the hydrogen, okay, to convert NAD to NADH. So here's a less fancy way of saying this. If you say the carbon compound is oxidized to reduce NAD to NADH, you would get that marking point there. Glycolysis gives me a net gain of 2 ATP. If you go on to explain like how that math works, that's cool, but it's not a separate mark. We produce 2 molecules of that 3 carbon compound called pyruvate. Sometimes you're going to see that written as pyruvic acid, samesies. All right, and then of course there are some other ways to write that, and that is A-OK. You're not going to see any marking points here for a diagram. That is okay. I still draw a diagram. You're going to have plenty of time. Draw the stupid diagram on the side, and that'll help you remember all of these things that you need to write about. Okay, how about this question? Describe the processes that occur in the mitochondria when oxygen is present. Well, it's kind of a dumb question because you can't get into the mitochondria if there's no oxygen present. They're just trying to confuse you. What they're trying to say here is describe the processes of aerobic cell respiration. Don't get confused. It's an 8-mark question. Just write everything you know about what happens in the mitochondria. So let's check out this mark scheme here. Pyruvate gets decarboxylated. So here's what you need to know here, that glycolysis happens in the cytoplasm, not the mitochondria. So you're not starting with glucose, you're starting with pyruvate. But anywho, so pyruvate gets decarboxylated, and that's going to give you carbon dioxide and NADH. That is a lot. So notice that they're saying both is needed here. So you're going to need to mention something about either removing carbon dioxide or decarboxylating pyruvate and reducing NAD to NADH. You're going to have to say both to get that mark. That 2-carbon molecule combines with coenzyme A to form acetyl-CoA. So honestly, if you just say that after pyruvate is decarboxylated, it's a 2-carbon molecule that combines with coenzyme A to form acetyl-CoA. That would get you that mark. Acetyl-CoA is what enters the Krebs cycle. 2 carbon molecules are going to be removed as waste. Let's see, that NADH molecule is formed. I would have said reduced, but okay. For each turn of the Krebs cycle, you're going to produce 3 NADHs and 1 FADH2, 1 ATP per pyruvate or per turn of the Krebs cycle. That's fine. And then those reduced NADHs or FADH2s are going to move on to the electron transport chain. So notice that this 8 marks includes talking about the link reaction. It involves talking about the Krebs reaction or Krebs cycle and covers the electron transport chain. And honestly, there's so much happening there. 8 marks shouldn't be that hard. So oxidative phosphorylation, again, is the method by which ATP synthase manufactures ATP. As electrons move along the electron transport chain, those hydrogen ions are going to be pumped into the intermembrane space. Gotta say inter, not inner, you won't get the mark. That creates a proton gradient. Another way of saying that is it creates an area of high concentration. You would get the mark there also. ATP is synthesized when they flow back passively through ATP synthase. Be careful with your verb here. It has to be something that tells them you understand that that process is passive. If you just say move, they might think that you think it's being pumped, so be very careful. I don't even think I would use the word flow. I think I would say move passively, just so that they're clear. And then ATP is synthesized by chemiosmosis. So you could talk about, again, how oxygen is the final electron acceptor, okay, to form water. I see here, award any of the above marks for a clearly drawn and annotated diagram. This annotation is the hard part here, so you're not going to get credit for just drawing a bunch of circles and arrows. You better be saying what compounds they are, and then you got to talk about things like oxidation and reduction. It's a whole thing. So again, my strategy is always to draw the diagram, and then in addition to write all this stuff, okay? So, yeah, it's a long mark scheme for this one. And then the final question we wanted to take a look at is explain how chemical energy for use in the cell is generated by the electron transport chain and chemiosmosis. So again, they're just being cheeky. They're testing you. All right, so they want you to just talk about the electron transport chain and chemiosmosis. When they're talking about chemical energy for use in the cell, that's their dumb way of just saying ATP. That's the chemical energy that the cell is using. Don't let them fool you. All right, so that energy is going to be carried by those NAD or FAD molecules, okay? So first we have to reduce them, so that's cool. You get a mark for just saying that NAD and FAD get reduced. Cool. That reduced NAD, so you can either say reduced NAD or you can just use its reduced form, which is NADH. That's cool too. Is produced in the glycolysis, link reaction, and Krebs cycle. It delivers electrons to the electron transport chain, which is located in the folds of the inner membrane called the cristae. As they get passed from carrier to carrier, it releases energy. And again, that electron at the end of the chain is going to get accepted by oxygen. Proteins act as proton pumps. Again, proton just means hydrogen ion. You're welcome to call it a proton or write it like this as a hydrogen ion. It's the same thing. They are pumped into the intermembrane space to establish a gradient. And then let's see, what is this one saying? So it's just saying, yeah, the energy from that electron is used to power those pumps. ATP synthase is an enzyme located in the inner membrane. And then let's see, as protons move down that concentration gradient, they go through ATP synthase, and ATP synthase is going to convert them to ATP. That's called oxidative phosphorylation. So if you did not get a chance to annotate your diagram with just me talking, please feel free to use some of these marking points as annotations for your diagram.